Friends, today we are going to discuss excretory system and physiology of excretion in human beings. As this topic is a bit large, so it can be explained in two parts. Okay, let us get started. The first part. You know very well about the excretion. Excretion is totally different from egestion. We need to understand the basic difference between excretion and egestion. Excretion is separation and elimination of nitrogenous waste materials from our body. Whatever metabolites that are formed in our body, if they are excess in quantity, or if they are not useful to our body, or if they are toxic to our body, they can be eliminated by separating them first from the tissues, and later they get eliminated by the parts of our body. That is actually called excretion. But what is egestion? Egestion is simply throwing out of the fecal material. Whatever food material that we have eaten is not digested, so such indigested materials are eliminated by a process called egestion by anus. So this is the fundamental difference between egestion and excretion. Excretion means separation and elimination of the nitrogenous waste materials from our body. You know very well, the separation is done from the tissues by a very important fluid called blood. Blood separates the excretory materials from the tissues and is transported to the excretory organs, and excretory organs separate the excretory material from the blood, and later such material is eliminated by using water or by not using water like that. Okay. Excretion is a process by which the metabolic waste is eliminated from an organism. So excretion in the case of human beings is carried out by lungs, by kidneys, by skin, and by rectum and certain other organs. In unicellular organisms, waste products are discharged directly through the surface of the cell because the unicellular organisms have no special organs of excretion. You may take, for example, amoeba. Directly through the body surface, the ammonia is eliminated. Of course, the excess of water in amoeba is eliminated by a special structure called contractile vacuole. So contractile vacuole is also useful for osmoregulation. Okay, through general body surface, in most of the organisms, lower grade organisms, excretory materials are eliminated. The main excretory products are carbon dioxide, which is formed as the metabolic end product of respiration, ammonia, urea, uric acid, and guanine, and also creatine. These are the main excretory products, but of which ammonia, urea, and uric acid are formed in large quantities. Is excretion also there in plants? The answer is yes. Plants also excrete the materials, but the plants store the excretory materials in their vacuoles. You take, for example, basil plant, lemon, eucalyptus. The excretory materials may be alkaloids in plants, or they are essential oils, or sometimes latex from Hevea brasiliensis, glucosides, cardiac glucosides maybe, tannins, resins, gums, organic acids, and mineral crystals. Of course, you know very well oxygen is also an excretory material in the case of plant photosynthesis because it is produced in excess quantities. The excess of oxygen which is produced during photosynthesis is eliminated. As it is eliminated, we need to say that it is also an excretory product. This is the basic concept of excretion. Whatever material that is formed in our metabolism which is produced in excess quantities than the required quantities, that shall be eliminated by a process that is called excretion. And plants produce as their excretory products several medicines to us. Such excretory products are useful as medicines to us. and they also produce certain drugs. Okay, right. Next, see the slide. I have collected it from the net. So this is the structure of uric acid. Uric acid is one of the excretory materials. Ammonia is also one of the excretory materials. Carbon dioxide, of course, eliminated by a process of breathing. And urea is the main excretory product of many terrestrial animals. Guanine is also an excretory product. Creatine, allantoin. So these are all the excretory materials. Okay. Friends, animals can be classified on the basis of their excretory materials. On the basis of their excretory materials, animals are classified into these many types. Ammonotelic organisms. By the definition, ammonotelic organisms are those organisms which eliminate their excretory product in the form of ammonia. Take for example protozoans, sponges, fishes. They eliminate their waste material in the form of ammonia. But remember one thing, that ammonia is a very toxic gas, or it is dissolved in water, but for the elimination of ammonia, lot of water is necessary. So the animals which are living on terrestrial medium, or the animals which are living in water scarce areas, cannot expend water for the purpose of ammonia elimination. So they convert ammonia into certain other non-toxic products. So ammonia is eliminated by aquatic organisms. The organisms which are living completely in water can spend a lot of water for the elimination of ammonia. That's why we can blindly say that the ammonotelic organisms are aquatic organisms. And next one, ureotelic organisms. Ureotelic organisms are those organisms which convert ammonia into less toxic urea, and urea is eliminated as their excretory product. What is the advantage? That the main advantage is that urea can dissolve in less amount of water. Less amount of water is needed for the elimination of excretory material. Of course, for the synthesis of urea, some energy is to be consumed, but water is a very important thing. That is why water can be conserved by converting ammonia into urea. And the examples are cartilaginous fishes, amphibians, mammals, etc. Certain amphibians, if you see the larvae are living in water and adults are living on land. So when the stage is in larval development, they eliminate ammonia, or they excrete ammonia. When adult stage comes onto the land, then their kidneys modify to eliminate urea. Of course, urea is synthesized in liver. The urodil larva, when it is there in water, it eliminates ammonia, and when it comes onto the land, so immediately it changes its products, excretory product, from ammonia to urea. So friends, just realize that the excretion of material, a particular product, or the excretory material nature is totally dependent on the availability of water. And if animals are having very little access to water, they cannot spend even some amount of water for their excretion. And such animals change their excretory product, modify their excretory product as uric acid. So uric acid needs very little amount of water, and it can be stored in body also, and it can be eliminated as a solid or semi-solid paste. Example of uricotelic animals are insects, reptiles, birds. Okay, and certain special animals like arachnids, the spiders, they eliminate guanine as their excretory material. They are called guanotelic. Okay friends, based on the excretory products, animals are broadly categorized into ammonotelic animals, example protozoans, sponges and fishes; Ureotelic animals, example cartilaginous fishes. Cartilaginous fishes store urea in their body fluid for bringing equilibrium to their body concentration with the salt concentration outside in seawater. So ureotelic animals, example cartilaginous fishes, amphibians, mammals. Uricotelic, example insects, reptiles, birds. Guanotelic, arachnids. Now, so how is ammonia synthesized in our body? What is the basic metabolism of synthesis of ammonia? When amino acids are used in our body as a part of metabolism, deamination occurs. As a part of deamination of amino acids, ammonia is synthesized. Okay, first, ammonia, the route of ammonia formation is through amino acid catabolism. Okay, the amino acids are subjected to transaminases enzymes. As a result of that, this amino acid will be converted into glutamic acid, and the glutamic acid, by the action of glutamate dehydrogenase, ammonia is formed. Ammonia can be synthesized from the glutamic acid. So this process where transaminases and glutamate dehydrogenase enzymes are useful. So this process is known as a transdeaminization. So transaminization occurs first. Okay, amino group is transformed from one position to the other position, and then it is subjected to the addition of hydrogen. That's why ammonia can be synthesized by a process called transdeamination. Free ammonia in tissues, when ammonia is formed in the tissues, it combines with the glutamate to form glutamine by glutamine synthetase. The glutamine is non-toxic material, and it is a non-toxic method of ammonia transport, because ammonia cannot be transported directly through blood or through any media because it is most toxic one. So that is why it is converted into less toxic material, that is called a glutamine, and a glutamine is transported. So the transport form of ammonia is called a glutamine. And you have seen protozoans and you have seen fishes. These are the animals that eliminate ammonia. Okay, why animals have chosen urea as their excretory material? The only answer is evolution of terrestrial animals. When the process of evolution of terrestrial animals occurred, they need to conserve water, because water is available only in the vicinity. So they are not living completely within water. So they need to conserve some amount of water. So as a result of that, ammonia is converted into urea in a special cycle called ornithine cycle in a very important organ called liver. So liver is an excretory organ because it synthesizes urea. Urea is less toxic when compared to the toxicity of ammonia, and it needs less amount of water to eliminate. It can also be stored, as I mentioned in cartilaginous fishes. Urea is stored for maintaining osmoregulation. So ammonium can be combined with the bicarbonate ions and aspartate. By utilizing 3 ATPs and water, it forms urea, fumarate, and 2 ATPs and AMP, like that. The interesting thing is urea requires 10 times less water for elimination when compared to the elimination of ammonia. That's why many terrestrial animals adapted urea elimination. Example, amphibians, mammals, chondrichthyes fishes. And the other very important excretory product is uric acid. So uric acid is the excretory material in those animals which are very little access to water. So they convert the urea into uric acid, and because uric acid needs 50 times less water when compared to the elimination of ammonia. So that's why uric acid is preferred as their excretory product in certain desert animals or certain animals which do not have any access to water, like that. Uric acid is also non-toxic material. It is insoluble in water and it can be eliminated in semi-solid paste. Okay. Uric acid contains purine ring with nitrogen atoms. These nitrogen atoms are derived from aspartate, glycine, glutamic acid. So, uricotelic animals are lizards and certain reptiles and then insects. Okay, what are the organs of human being? And we say that organs of human being are kidneys. But so other than kidneys are only separating the material from the blood, separating the excretory material from blood and eliminating. But from the ammonia is transported and where is it converted into urea? So that is also considered, that organ is also considered as an excretory organ. Okay, and not only urea is the excretory product. There are several other excretory products produced in our body. That may be lactic acid, or carbon dioxide, bile pigments, and certain drugs what we have taken during fever, and several other materials can be eliminated. Let us see skin. Skin is called jack of all trades but master of none. It is doing almost all many functions in our body. It gives protection, it gives support, it gives shape to our body, and it is responsible for thermoregulation. It is responsible for osmosis also, and for several reasons, for the synthesis of vitamin D like that. So skin is jack of all trades, and it is also an excretory organ because it eliminates uric acid. It eliminates lactic acid and it also eliminates urea. Coming to lungs. Lungs are the main respiratory organs. They are the organs of the exchange of gases. The respiratory gases are oxygen and carbon dioxide. Oxygen is taken in and carbon dioxide is eliminated at lung surface. The exchanged carbon dioxide is sent outside or thrown outside or eliminated outside through the respiratory tract. Okay, right. Carbon dioxide is also an excretory material. In the case of amphibians, carbon dioxide is mainly eliminated by the gateway that is called skin. Okay friends, liver. Liver is a very important metabolic center in our body. Often it is described as metabolic brain. Okay, what does it do? So it forms or it produces urea. The main excretory product of our body is urea, is synthesized in liver. That's why liver is considered as the main excretory organ where urea is synthesized in ornithine cycle. Not only that, liver can separate the bile pigments which are the products of RBC destruction. When RBC are destroyed and hemoglobin is subjected to some metabolism, bile pigments are produced. These bile pigments, if they are mingled with the blood or if they are entering into the tissue, they become toxic. That's why very carefully liver separates them and eliminates directly into the digestive tract through bile juice. So from the digestive tract it is eliminated or digested through the fecal material. The fecal material of a species is having a characteristic color. The color of the fecal material of any species is because of their bile pigments. The bile pigments may be bilirubin or biliverdin. Okay, bilirubin is golden yellow in color and biliverdin is green in color. Okay, friends, large intestine, the rectum especially, where lot of digested material is stored, and from the large intestine, drugs. If any drug that we have taken and is not metabolized in our body and it is eliminated by means of certain very special secretory cells, epithelial cells of the large intestine or the rectum. So rectum eliminates drugs from our body, And today's topic is the nitrogenous waste material elimination that is performed by the kidneys. Friends, let us see the gross organization of human excretory system. The gross organization of human excretory system. This system is consisting of the main organs such as kidneys. You see the diagram here, kidneys, ureters and urinary bladder and also urethra. Okay, very simple excretory system. Kidneys, ureters, bladder, urinary bladder and urethra. You know very well about our kidneys. The kidneys, there is a pair of kidneys situated on either side of our vertebral column in the lower part of our abdomen, attached to the dorsal wall. Okay, attached to the dorsal wall by certain mesenteries. And friends, you know that kidneys are having bean shaped. Outer surface is convex and inner surface is concave. They are bean shaped. They are very well protected by a capsule called renal capsule. And lot of fat also got deposited. Because of this fat or the adipose tissue, so lot of impact because of mechanical disturbance can be reduced. Okay, mechanical shocks can be absorbed by the fat that is stored nearby the kidney. Okay friends, if we see the internal anatomy of the kidney by taking the LS of kidney, longitudinal section of the kidney, we can understand everything. So kidney is having two special tissues. Outer part of the kidney is called cortex and inner part of the kidney is called medulla. Okay, the cortex and the medulla are the two histological regions. Cortex is situated in the peripheral region and the medulla is situated in the central region. Exactly in the central region, just beneath the medulla, there is a very big cavity or very wide open space is there. This space filled with the renal fluid is known as pelvis. Okay, let's see the cortex. This is called cortex. It is situated in the peripheral region. Now, just beneath the cortex, there are several pyramid-like structures. These pyramid-like structures are 1, 2, 3, 4, 5, 6, 7. There may be 8 to 15 pyramid-like structures. And these pyramid-like structures are called renal pyramids. And they form medulla. Pyramids form medulla and outer part forms cortex. In between the pyramids, cortex is projected. That projected part is called columns of Bertini. Columns of Bertini. Okay. What are there in cortex and in medulla? In cortex and in medulla, the fundamental units, structural and functional units of kidney are there. And these are called nephrons. Because the nephrons are the structural and functional units of kidney, the study of kidney is called nephrology. It is called a nephrology because every action of kidney or every function of kidney is performed by its fundamental unit called nephron. Okay, the nephrons may be situated at the junction between the cortex and medulla, juxtamedullary nephrons, or some nephrons may be located deep into the medulla. Their Henel's loop is deep into the medulla. They are called medullary nephrons. The cortical nephrons that are situated at the junction and juxtamedullary nephrons whose Henle's loop is situated deep inside the medulla. The medulla at its conical projection, inner projection, are having small pores. Okay, these are called renal papillae. These renal papillae open into the funnel-like structures. The funnel-like structures here, you see here 1, 2, 3 like that. These funnel-like structures are called calyces. The calyx which is small is called minor calyx. Minor calyces open into a very big funnel-like structure called major calyx. the major calyces, 1, 2, 3 like that, open into a very, very wide space that is called pelvis. The pelvis comes out through a very big aperture of kidney that is known as a hilum or a hilus through in the form of a duct. That duct is known as ureter. Okay, so this ureter, left side and right side, the two ureters move towards posterior side and finally open into a bladder. That bladder is known as urinary bladder. Okay friends, this is the structure of kidney. But what is very, very important here is kidney histologically is having nephrons. Okay, but in between the nephrons there is interstitial tissue. The interstitial tissue, which is situated between the nephrons, among the nephrons, is very, very, very important because it is having a gradient of osmolarity. The concentration is different in different parts of the kidney interstitium. In cortical region, that means in the peripheral region, the concentration is less. As you go deeper and deeper into the medullary region, concentration of interstitium gradually increases. Osmolarity, okay, osmolarity gradually, milliosmoles per liter we measure. So that osmolarity gradually increase. The gradients of osmolarity from cortical region to the medullary region is a very, very, very important physical factor for the reabsorption of water and making the urine much concentrated. Okay, so there I mentioned the external morphology. There are two kidneys and each kidney is having the length of 10 centimeter and having the width of 6 centimeters, and the kidney is having a thickness of 4 centimeters. 10 centimeters length, 6 centimeters width, and a thickness of 4 centimeters. And each kidney is having 1.2 millions of nephrons. 1.2 millions of nephrons. And each kidney is having a weight of 200 to 250 grams. Okay. If you see the left diagram here, okay, there are two kidneys, a pair of kidneys. This is called, right kidney this is, and this is the left kidney, wrongly here written as a left kidney, but this is right kidney. Okay, this is right kidney and this is left kidney. Right kidney is smaller, left kidney is larger. Okay, in this diagram I have wrongly written it as a left kidney, but it is right kidney. This is left kidney. Okay, left kidney is larger, right kidney is smaller, so kidneys are slightly asymmetrical. Why this is small in size? Perhaps above this there may be a very big space occupied by liver. That may be the reason. Okay friends, this is a right kidney, this is a left kidney. Okay, a small mistake is there in the diagram. Now, okay, how many nephrons are there? 1 million to 1.2 millions of nephrons are there, and the nephrons are playing a very, very, very important role in the elimination of waste materials and in the reabsorption of water. One more speciality in the kidney of human beings, especially in mammals and also in birds, is that the nephron, the renal tubule of nephron, is having a hairpin bend. The hairpin bend is called loop of Henry and it is characteristic, and because of this arrangement lot of water, salts get reabsorbed. That is the very important characteristic arrangement of the nephrons. How is a nephron organized? Let's see. Nephrons are the structural and functional units of kidneys. Each kidney, as I mentioned, is having 1 million to 1.2 millions of nephrons. And let us see the structure of nephrons. Each nephron is having two fundamental regions. One region is called a Malpighian region or Malpighian corpuscle, and the other region is known as renal tubule. Okay, This is called a Malpighian body or Malpighian corpuscle or renal corpuscle, and the second part is called renal tubule. This is called a very, very long renal tubule. Okay, what are the parts of Malpighian capsule or renal capsule? Okay, this area, Malpighian body, is consisting of two further parts. One is called a cup-like structure. The cup-like structure is called Bowman's capsule. It is called Bowman's capsule. And inside the Bowman's capsule, there is a ball of capillaries. This ball of capillaries is known as glomerulus. Ball of capillaries is called glomerulus. Okay, friends, if you can observe the glomerulus carefully, blood comes into the glomerulus through a very small arteriole, and blood leaves of this glomerulus by another arteriole. So this is incoming arteriole. Let me go to previous slide. Okay, this is incoming or incoming blood arteriole. This is outgoing. Incoming one is called afferent arteriole, and outgoing is known as efferent arteriole. If you can carefully observe, afferent arteriole is having a larger diameter and efferent arteriole is having smaller diameter. The diameter of efferent arteriole is smaller and that of afferent arteriole is larger. And a ball of capillaries is there, and this capillaries is called glomerulus that is fit in the Bowman's capsule. The Bowman's capsule is a cup-like structure having two layers of squamous epithelium with podocytes. This area, that means the entire Malpighian body, is the main area for filtering the blood by a process called glomerular filtration. Okay, next let us see, this is Malpighian body, and the next one is called renal tubule. Okay, renal tubule. This renal tubule is very, very special and characteristic. It is not straight, it is bent in the form of a hairpin. So that is why it is very special, and it is regionalized into different parts. The first part, let's see, the first part of the renal tubule, what has just come out of Bowman's capsule, is called neck, and this area, the next part, got lot of folds. So this area is known as a proximal convoluted tubule. Proximal convoluted tubule. Proximal convoluted tubule inside are having glandular epithelium with a brush border, indicating that this area, that is proximal convoluted tubule, is the very important area, significant area, very critical area for the reabsorption of almost all 85% of the renal content, renal fluid, because it is having small microvilli in the form of brush border. And then the PCT, that means proximal convoluted tubule, opens into loop of Henle. The loop of Henle is having two limbs. One is called descending limb, the other one is called ascending limb. The descending limb and ascending limb make a hairpin bend here. This is very, very important characteristic structure of the mammalian nephron. The descending limb is anteriorly thicker region, and later it will become thin. How is descending limb moving? You see, Bowman's capsule and glomerulus are located in the cortical region. This is cortex, and this part is located in the medulla. So the renal tubule is, especially the descending limb, is going down into the medulla, because that is why it is called descending. And after taking a hairpin bend, ascending limb is coming towards cortex. Okay, moving towards medulla by the descending limb, and coming towards cortex by the ascending limb. And such a kind of arrangement is helping to reabsorb more water and some very important salts by means of counter current mechanism. Okay, this is descending limb and this is ascending limb. Ascending limb is having more thicker area because ascending limb is impermeable to water. Descending limb we discuss in the physiology of urine formation later. And let's see, the descending limb is permeable to water. PCT is also permeable to water. The permeability of water is because of some small pores. They are called aquaporins. Aquaporin 1, that means small pores that allow water, that do not allow, such pores not allow the other solutes. They allow only water. So more aquaporin 1 type 1s are there in PCT and descending limb. Such aquaporins are totally absent in this ascending limb. Friends, in the year 2003, the Nobel Prize was awarded to the person who described these aquaporin structure and their function. Okay, this is descending limb and this is ascending limb. The ascending limb, after reaching the cortical region, is again convoluted, and this convoluted portion, second convoluted portion, is called distal convoluted tubule. The distal convoluted tubule inside is also having simple glandular epithelium, but here brush border is not there. Principal cells and intercalated cells are there, and potassium absorption, potassium secretion, bicarbonate secretion is a very important aspect here. Secretion and absorption of potassium bicarbonate to maintain acid-base balance is a very important component here. And this DCT is also being influenced by ADH hormone. ADH means anti-diuretic hormone or vasopressin. Here there is a very important organ, if you can locate, between the afferent arteriole and then the distal convoluted tubule. There is an organ. This organ or apparatus is called a juxtaglomerular apparatus. The juxtaglomerular apparatus is significant in secreting a hormone called renin that will maintain the GFR or reabsorption rate. Okay friends, this is distal convoluted tubule. And after that, a small junction tubule is there. This junction tubule attaches or joins the distal convoluted tubule with a collecting duct. This is called a collecting tubule or collecting duct. This collecting duct is also moving from cortical region to the medullary region. Cortical region to the medullary region. Okay, and it is receiving so many junction tubules of other nephrons. So this is called the structure of a nephron. Okay, the structure of a nephron, after having understood, we need to understand the absorption process. Okay, microvilli in PCT absorb sodium and other materials because they are having symporters for glucose absorption, amino acid absorption. The absorption may be secondary active transport or primary active transport. Primary active transport is for sodium and secondary active transport is for glucose and amino acids. Okay, and then descending limb is highly permeable to water, whereas ascending limb is impermeable to water. Descending limb is having aquaporin 1s. Aquaporin 1 can conduct what is that called obligatory water reabsorption, and aquaporin 2s are there. Aquaporin 2 are there in distal convoluted tubule, where facultative water reabsorption is there. This is facultative water reabsorption, and here mandatory water reabsorption occurs because of their components. Okay, right. So a nephron is having a network of capillaries. You see network of capillaries around PCT and DCT, that is called peritubular network. And a network of capillaries is there around the loop of Henle. This is called vasa recta. Vasa recta and the loop of Henle are having the fluids which are running in opposite direction. So these kinds of opposite running fluids always maintain certain osmolarity in the interstitium of kidney, so as to absorb more water. So this is the structure of nephron. As I mentioned, there are two types of nephrons. Some nephrons are situated at the junction between cortex and medulla. They are called cortical nephrons. And many of the nephrons, some of the nephrons are located, okay, some of the nephrons are located in what is that called, more deeper in the medulla, whose Malpighian body may be situated in the cortical region. So such nephrons are called juxtamedullary nephrons. Juxtamedullary nephrons are only 15%, whereas the cortical nephrons are 85%. So many number of cortical nephrons are there, and only 15% of juxtamedullary nephrons are there. And there are some differences between the cortical nephrons and juxtamedullary nephrons. Let us see. In cortical nephrons, the glomerular apparatus is situated, the glomerular apparatus or glomerulus is situated upper in the cortex region. Whereas in the juxtaglomerular nephron, the glomerulus is situated at the junction between the medulla and cortex. Okay, position of glomerulus is different between these two. Position is in the cortical region, glomerulus position is in the cortical region, glomerulus position is at the junction. Okay, now the glomerulus in cortical region is, cortical nephrons is small, the glomerulus is large. Okay, the loop of Henle is situated up to the upper part of the medulla only in cortical nephrons, whereas the loop of Henle is very long and is situated deep in the medulla. Okay, so the what is that called, the extension of loop of Henle, there is a difference. The size of glomerulus, there is a difference. The location or the position of the glomerulus, there is a difference. Okay, now you see the ascending limb is thicker. Here ascending limb is thin. In cortical region, cortical nephron, ascending limb is thicker, and in the juxtamedullary nephron, ascending limb is thin. And in cortical region, the cortical nephron, the rate of filtration is very, very slow. The rate of filtration is very low because glomerulus is very small. Here, the rate of filtration is very high because glomerulus is large here. That's why rate of filtration is very, very high. Cortical nephrons are not meant for concentration process of making the urine concentration. So they are simply for reabsorption of more materials. So because the loop of Henle is not situated deeper in the medulla, there is no osmolarity gradients or concentration, so they are not for urine concentration. But juxtamedullary nephrons are mainly meant for urine concentration. Here, the efferent in the cortical nephrons, efferent arteriole forms peritubular network. Here, the peritubular network is forming the is having a connection with the vasa recta. Okay, right.